This is a work presented by the Canadian Bioinformatics Workshop. This work is under a Creative Commons license. Here we come with the final slide for the Pathway and Network Analysis Workshop, and we are going to summarize pathway and network analysis using different omics data. So we have seen during this workshop 2 ways of using pathway. So the first way on the left is to use pathway as pathway database to do pathway enrichment analysis. And we have seen how to visualize this pathway as a network where the nodes will be the pathways and the link between the pathway will be the genes in common. And on the right, we have used the information of a pathway to create a functional interaction network where the network would be, the nodes will be the genes and the edges would be the functional interaction between the 2 genes. So, but now sometimes it's confusing and we don't know how to use when to use an enrichment map, a Reactome FI network, or a gene mania. So the way I do it is I try to see the size of my gene list to decide which network I need. So if I start with a very large gene list, let's say more than 100 genes, until 2000 genes, well, I know that I really need to summarize by pathways. So in this case, I will do pathway enrichment analysis followed by the visualization of the pathway result using an enrichment map. So in this network, the nodes will be the pathway and the edges will be the gene in common. But what about if I have a medium size, let's say 100 genes, maybe I will be able to for sure summarize as pathway first, but also to represent as a network of genes, for example, using the Reactome FI app. So in this case, my network, the nodes will be the gene and the edges will be the functional interaction between my 2 genes. In this case, because my network is going to be very big, like 100 genes, I don't want to add linkers. So I'm not going to add additional gene to the network. I just want to extract the cluster of genes that are connected with each other, because then maybe I'm going to identify some kinds of protein complex or at least proteins that work in the same function, and I may discard the genes that are not connected to these complexes. Then if I have a small gene list, let's say 1 to 50 genes, then I don't need to summarize by pathway. So what I'm going to do is I'm going to try to expand the list using function prediction. So the recommended tool for this is Gene Mania. Also Reactome FI with linkers, but it's more Gene Mania if you want to add related genes in terms of multiple attributes. So in this case, so the network is, the nodes will be, in this network, the nodes will be genes, and it will be linked by different functional networks. So here is the summary of the workflow that we have seen during this workshop. So first we have collected our genomics data, and we will see in the next slide that they can have diverse origin, but at the end we have a gene list that we want to functionally interpret in an unbiased way. And beside this, we have the collection of pathway database that give us the information about the function of these genes. Then we use pathway enrichment analysis to see the pathway enriched in our gene list, and then we can visualize as a network. We visualize as a network because it's easier to distinguish the relationships between the pathway or the gene in our list. And then the power of the visualization as a network in Cytoscape is that we can add multiple layers of information on one network. For example, we can add micro RNA targets information, transcription factor targets information, or drug targets. If you have a gene-gene network, you can use the color and the shape and the size of the nodes to indicate the value of the gene expression. So this is a roadmap, which is the same as the summary, but a little bit more detailed. And you can see in the blue rectangles the different sources of the gene list. So you can work with transcriptomics data or proteomics data, which are about the same similar pipeline, but you also can work with DNA methylation, microRNA, ChIP-seq, or SNPs. So the first processing step is going to be very dependent on your omics experiment, but at the end you are going to have a gene list that you would like to functionally interpret, so the downstream tools are going to be very similar. And then on the left here, in that identify interesting pathway is when you want to summarize using pathway enrichment analysis, and identify interesting networks on the right is when you want to create functional interaction network. Once you have done that, then you want to really understand your results, and then you look at the genes that are involved significantly in each of these pathway. You try to find a mechanism, and then you need to validate. You need to go back to the wet lab and validate the prediction performed by the bioinformatics tools. So now, so we talked about a lot about the transcriptomics, but what about you have microRNA? So let's say you have two conditions, treated and normal, and you have extracted your microRNA. So you have a list, sometimes a very large list of microRNAs that are differentially expressed in your condition. So here, the mirrors, they will not be included in all the pathway database. So you need to, if you want to do an enrichment analysis of these microRNA results, you need to find that pathway database containing microRNAs in the pathways. So there is some tools that are specifically built with this microRNA pathway database. So the pathway are organized into signaling pathways. For example, this one, and you can directly do the enrichment analysis on this pathway. But in other cases, maybe you have not done a microRNA experiment, but you have 2 or 3 microRNA that are the top hits of your omics experiment. And what you would like to do is to retrieve the targets, so the gene that are the targets of your microRNA. So you can use a specific database like the Diana Mirtar-based database to retrieve these targets. And for example, here, as shown is the enrichment map, you can overlap this microRNA targets to the pathway to see the pathway that contains a lot of these targets of your microRNA. So this is 2 ways that you can use to study your microRNA. Now, what about if you have metabolomics data? It's the same as the microRNA. What you have is metabolites, not genes. So if you want to do pathway enrichment analysis, well, you have 2 choices. The first one, A, is that you look at your metabolites that were up or down regulated in your experiment and try to find the enzymes responsible to produce these metabolites. And then you look at the enzymes and see if they are up or down regulated. The second way is to use a pathway database containing only the metabolites in the pathways, and then do your pathway enrichment analysis on this. And there is a tool, Metabolomics, that can contain this pathway database. So you can use this tool directly with your metabolomics data. And it's a very complete tool, so that can do also the pre-processing as well as the pathway enrichment analysis using a Fisher's exact test if you have a list of metabolites, or you can rank all the metabolites using GACA, and also doing some visualization for you. So it's a very complete tool, and I recommend it if you have metabolomics data. And I think there is also CBW workshop on this topic. Now, what about if you have GWAS data? So if you have GWAS data, you have some SNPs, and you need to associate the SNPs with some genes. So you can do it on your own, but there are also tools that are specific for GWAS that takes into account the linkage disequilibrium, and one that I've tried myself was Magenta, and I like this tool a lot. Maybe it's not very recent. So recently I found this Bridge published in 2024. Seems to be very interesting. I haven't tested myself, so please look at the publication and try it on your own. Then what about if you have ataxic or ChIP-seq data? Then, so what you have is a list of peaks or a list of open chromatin regions, and usually the output is a bed file with 3 columns. So the first column would be the chromosome names, then the second column would be the start of the peak, and then the position of the start of the peak, and then the third column will be the end, so the position of the end of the peak. So now what you have with this chromosome region is that you need to retrieve the genes associated with these peaks. So you can do it yourself using some R package, using some annotation, but there is also a tool, GREAT, that is specifically designed to do that, so retrieve the genes near your peaks using some rules that you can define to limit the boundaries upstream and downstream the TSS of your genes, and then do the pathway enrichment analysis. And then when you have this output of GREAT, then you can upload it in enrichment map because it's compatible with enrichment map. So we talk about GREAT, but there are other tools that exist that you can look, that Poly-Enrich and Lola. And there is also MEME and Homer. So MEME and Homer they have a slightly different goal because they are going to look at the gene list and the promoter of your gene list and look at enrichment of DNA motif. So it's also very interesting, but it's different from pathway enrichment analysis. Now what to do if you have single cell RNA-seq? So we saw it in module 6, but I'm going to repeat here again. It's very, very similar to the bulk RNA pipeline. So you do all your processing. Usually at the end, you visualize your result as a UMAP. So you will have your cluster that sometimes you annotate, and then you can follow the pipeline of bulk RNA-seq. So what we do usually is use Seurat file markers to find the top genes that are specific of each cluster, and we use it for annotation. So we use the top genes or the top pathway to give us a clear idea of the cluster, and then we can do an annotation of this cluster. Once our cluster have been annotated, maybe we want to do differential expression of this cluster between the condition A and the condition B because we have treated and controlled samples. So in this case, we aggregate our cells into pseudobulk, and then we follow the pipeline for bulk RNA using differential expression using tools like HR and DSEQ2. Then we have a robust rank file, and then we can use GACA and visualize into cytoscape enrichment. If you have like a gene list specific for your cluster, so one of your cluster, you also can take this gene list and visualize as a network of genes using, for example, GeneMania, Reactome-FI, String app. Our regulant that I'm mentioning here is an app that will take all genes in your gene list and will try to find the master regulator as of this gene list, or maybe a transcription factor that potentially regulate your gene list. So now if you work with non-model organism, then you need to find a pathway database for this non-model organism, or you need to find a pathway database of the non-model organism, which is the closest of your model organism. So you can do some phylogeny analysis and see which organism is very close to yours, and then you basically need like a pathway database in the type of like a GMT file. And for example, GProfiler has a lot of GMT file that you can download and use, for example, in GACA with your own data. There is also a script here to create a GMT file from the gene ontology using your gene annotation from your model organism. Also, Gary Bader's team is developing like a web version of enrichment map, so you don't have to download Cytoscape. And really, it's the same as the JMania web app, where you open a web browser and then you can input your rank file or your matrix of count. This is the first release of this app. It will be further improved, but you can test it using this link. Now, also for this last slide, we would like to remind you that Cytoscape has a variety of apps that you can find on the wall of apps. You can look at the description and find the apps that are convenient for your project. And it's time to get hands-on. So we are going to offer you a multiple optional assignments for this week. One using AttackSeek, ChipSeek. One is the integrated assignment that review all the pipeline and all the tools that we have seen during this workshop, and some script where you can do everything that we have done using gprofiler, GSEA, Cytoscape enrichment map, but using a script so you can reproduce your data in a more easy way when it's needed. So thank you very much, and we hope that you enjoyed the workshop.